Okay, so chapter 17 is about protein synthesis. So chapter 17 review protein synthesis. So you should review the beetle and Tatum experiment. This is with the bread mold. They basically mutated genes to figure out what they did. So they did bread mold. One gene, they came up with what they figured out is that one gene, one polypeptide hypothesis. So what this means is that you have a series of reactions, A, B, gets converted to C to D, and there's a different enzyme that catalyzes each one of these. And these enzymes are proteins, so that organism to make these three enzymes has three different genes somewhere in its DNA. Gene 1 makes enzyme 1. Gene 2 makes enzyme 2. So every polypeptide every gene makes a different polypeptide. All right, so that's what they figured out. All right, so then also we did an overview of protein synthesis. So we're going to look at prokaryotic cells versus eukaryotic cells. Prokaryotic cells do not have a nucleus. And so in this case here we have DNA in this cytoplasm. So this is in the cytoplasm. DNA gets converted to mRNA, which then gets converted to your polypeptide chain. So then what are these processes called? This one is transcription. And this one is translation. And this all occurs in the cytoplasm. And I should say that this occurs at the translation occurs at the ribosome. I didn't draw that in. At the ribosome. And so converting mRNA into a polypeptide. And so prokaryotic cells have one less step than eukaryotic cells. So let's draw the eukaryotic cells, nucleus, and cytoplasm. So in eukaryotic cells, they have their DNA in the nucleus. You make your pre-mRNA strand, and that is transcription. So that's in common with the prokaryotic cells. And then you have, a couple of things happen with your mRNA to get your final mRNA that is going to leave the nucleus. And so what is that final mRNA? What happens from here to here? That is called RNA processing. And so what happens with RNA processing? You cut out the introns, hence it's shorter, and you add a cap and a tail. The cap and the tail is going to help it to leave the nucleus and meet up out here with the ribosome. So here's your mRNA, the cap and the tail, and remember then you're going to have your growing polypeptide chain. And this process that's occurring out here at the ribosome is translation. So a couple of things different in prokaryotic versus eukaryotic cells is the addition of RNA processing. Prokaryotic cells do not have that because they do not have introns, so there's nothing to cut out. And so therefore, there's that extra step, and then also the compartmentalizing, that these guys happen in the nucleus and this happens outside in the cytoplasm. All right, so that's another big difference between them. For this chapter, also in the first part of it, the review part of it, you should look at the differences between DNA and RNA. So So how is DNA different than RNA? The bases, DNA is double-stranded, RNA is single-stranded, and you should know how to use that chart where mRNA is quote-unquote red in groups of 3 called codons. So your mRNA is read like this, like AUG, then UUU, and it's the mRNA that you use in the charts, the codons, not the DNA, not the tRNA. Okay, so let me turn the sheet over here. And so let's go through the overview of protein synthesis. So let's look at a little bit more in depth than this. So that was more of an overview. Let's do more in depth. Okay. So protein synthesis. Okay. So I'm going to go ahead and kind of draw here. I'm going to draw. I go like this, where this is the nuclear, so this is the nucleus, so this is the cytoplasm on this side. And this is the nucleus. All right, inside the nucleus. And so we're going to look at transcription and translation here. And so, so here's your double-stranded DNA. This is your DNA. And remember that there's a promoter region before the gene. So the promoter region, remember, has the TATA box. So the other side would be this. And then you have your gene. So let's say it's TACAAA. So the other side would be ATG. TTT. All right, so let's say that's the beginning of your gene, and I'll just put dot, 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 dot. All right, so you get the idea. So when transcription happens, what occurs? All right, so I'm going to do this here. All right, so here's your promoter region still. TATA. Whoops. I'm writing TATA again. ATAT. All right, so this is still your promoter region, and this is your gene, so your TACAAA, and over here is ATG, TTT. All right, so what happens is, what's the significance of the TATA box? It's to bind the transcription factors. And then that's how RNA polymerase knows where to bind. So let's do a different color here. Here's RNA polymerase. And RNA polymerase is going to use just one side of the DNA. So let's say we're going to use the top side. So then it's going to move along and add the RNA nucleotides. So we're going to have A, U, G. All right, that's the first one. Then U, and so on. So you're going to build that second strand. And it's built in the 5' to 3' direction. So that means that this is the 3', this is the 5', which means what must be true about this DNA here? This must be the 5', and this is the 3'. So if it's going to move in this direction here, because that's where the TATA box is, it's going to move and make your mRNA strand. You're not going to use this side. This side would be the 3', this would be the 5' end. Not going to use this side. mRNA is only single-stranded. Actually, let's go ahead and put in, to make it more similar to your notes here, let's go ahead and add some. Let's say that this is CCC and then ATT. So let's add some bases, a couple of things on here so I can show you here. So I want to show you here, so it's opened up here, but I want to show you that it opens up in a bubble. So I guess that's what I want to, all right, show you. So then this would be CCC, then ATT, and then this would be GGG, then TAA. So it's opened up in a bubble like this. And so this will continue on and build GGG, and then this will close up, and this will open up, and it'll add UAA. So it'll continue moving on in that 5 to 3' direction. And so then what I want to show is then when you're finished making your mRNA, so this is what we have, here's your mRNA, you're going to have in the 5' AUG, then UUU, then GGG, then instead of not T, it's going to be UAA. All right, and then this is the 3' direction. So this is called your what? This is your pre-mRNA. Why is it pre? Because it is needs to be processed. All right, so let's go through the processing. So what are the 2 steps for processing? It is cutting out the introns and also adding a cap and a tail. All right, so if we look here and we treat, let's say we treat this as an intron right here. When we cut it out, let's say the rest of them are exons, so we're going to cut that out, so we're going to have a shorter mRNA strand. We're going to have AUG, then GGG, then UAA. GGG. 3'. All right, and so this is your final mRNA. And this process is the processing. All right, so this was the transcription, making the mRNA. This is the processing. And so, so we can say RNA processing. What's the thing that I haven't added into processing? Is adding your cap and tail. So add the cap and tail on either side, and we're done with processing. So now this will leave the nucleus through a pore. So this will leave the nucleus and go to where? Where it'll meet up with a ribosome, but what part of the ribosome? It's going to meet up with the small subunit. So this is the small subunit of the ribosome. And here's your mRNA. So you're going to have AUG, GGG, UAA. All right, so it meets up with that. Then AUG is the start codon. So there is a tRNA molecule that has the correct anticodon. So you have a tRNA molecule, just like this, has the anticodon UAC. And that tRNA only picks up one amino acid, and that is methionine. If you look at AUG on the chart, it stands for methionine. That's because the tRNA UAC comes and binds to it, and that first amino acid is methionine. Next comes in your large subunit. So your large subunit now binds to this. Large subunit has 3 sites. The E, P, A. E is just the exit site. So now your first amino acid is in the P site. So now that leaves the A site with the second codon open. So therefore there's a tRNA that has CCC attached to it. And how do I figure out what amino acid is going to be hooked there? You use the chart. All right, so here's the chart, and we use the codon of the mRNA. So GGG, so we go G, 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 and that's G is GLY, which is glycine. So this is going to come over here, CCC, GLY. All right, so then what happens next is, let's get a different color here. We have to hook these two together so this bond is broken. this bond is formed, and this little guy is going to leave. So then, if we look at this picture next, so then if I'm going to redraw this over time, so E, P, A site, and here's your mRNA. This, remember, the ribosome moves down a codon, so AUG is now in this E site, GGG is here, UAA is here. All right, and so let me draw the mRNA strand. All right, 5, 3 prime. So now you have this tRNA with 2 amino acids here, CCC, hooked to glycine and methionine. Okay, so that's what we have here. This tRNA has done its job. So this little guy here with the UAC, nothing attached to it anymore. That leaves, all right, or exits, hence the E site. And now we have our growing strand. So now we have our next codon in the third spot. The next codon is UAA. If you go and use your chart, UAA is a stop codon, so it doesn't code for an amino acid. So I did this specifically here. This would continue on and on and on by doing the same process that we just did here. I just made it short for review. And so what comes and binds to that is a release factor. So a release factor binds here at the stop codon. And what does that trigger to happen? Your 2 amino acids leave. All right, so therefore you have your met and your glycine. So it's very, very short, dipeptide there. And mRNA comes off, large and small subunit dissociate, and you have your finished polypeptide chain. And that is protein synthesis. Now, the last bit with that chapter was that you need to review is your mutations. So when mistakes occur, what happens? So there were base pair substitutions. So like silent mutations. Missense and nonsense. And then there were insertions and deletions. So there is frame shift. Shift causing nonsense, frame shifts causing missense, or insertion or deletion of whole codons. Okay, and so that was the end of chapter 17. So review those types of mutations and look at the severity. Like for instance, insertions and deletions usually cause a lot more change than some of the other ones, especially a silent mutation. So review that. All right, and that is chapter 17 in a nutshell.